Welcome to the second part of lesson 3.1. In the first part, we've discussed bacteria, what they are, as well as their essential role in nutrient cycling. So now we are going to look at the specific different types of nutrients, the essential nutrients for plants and for other organisms that bacteria are able to gain from the soil matrix, the silt, sand, and clay, and also from the atmosphere as seen in the nitrogen cycle. So first of all, we'll talk about the nitrogen fixing bacteria. Then we'll move on to the phosphate solubilizing bacteria or phosphate solubilizing microbes, as bacteria are not the only ones that are capable of doing this. And then we'll look at sulfur solubilizing bacteria, and then finally potassium solubilizing bacteria. So these are all macronutrients that only really bacteria and fungi can actually access for the plant. And this is why it's so important to talk about these macronutrients and how this process occurs in soils in order to understand the importance of biodiverse community of these microbes being present in soils in order to restore our soil ecosystems. All right, without further ado, let's look at the nitrogen cycle. This cycle in particular is very important. It's so important and it's so difficult to obtain nitrogen from the atmosphere, which is really abundant. It's the most abundant atmospheric gas, but it's very, very difficult to break those three bonds that are present between the two nitrogen atoms. And really the only living organism that can do this is bacteria, so as far as we know. They have an enzyme that is able to break down this bond, and nitrogen is extremely important in all kinds of cellular functions. It plays an integral part in chlorophyll, amino acids, proteins, adenosine triphosphate, which is ATP, is actually a molecule that our body uses or cells use. All cells use really to exchange energy. It's an energy containing molecule. And also in things like DNA and RNA. So a very genetic expression depends on nitrogen being present. There are both symbiotic nitrogen fixers and free living. So these bacteria divide into these two groups, and we'll actually go into that in detail. In most undisturbed soil types, atmospheric nitrogen is turned to ammonia. So this is a nitrogen cycle, and I'll give you a few examples just to grasp this concept. But atmospheric nitrogen has to be split by nitrogen-fixing bacteria, living in this case in legume nodules. So little nodules are created by the plant through the bacterial stimulation, and the bacteria hide in there and will do the splitting of the dinitrogen. And this nitrogen can be fed to the plant, so straight away the nitrogen goes to the plant, or through the process of ammonification, it is converted into ammonium ion. Then it can also be nitrified by nitrifying bacteria into nitrites, so this compound here. And nitrifying bacteria can further create nitrate from nitrite. And this can also be assimilated by the plant. So these compounds, these molecules, can actually be used by other plants that have not got nitrogen-fixing bacteria. So it's a very nice way of sharing nitrogen from the atmosphere across the soil community through these different groups of bacteria. Now, denitrifying bacteria, when oxygen conditions are reduced, they will actually release nitrogen as gas back into the atmosphere. This is not something we want. We'd prefer that once the nitrogen is fixed by nitrogen-fixing bacteria, it can cycle throughout this process and be locked up in the organisms. So the other way nitrogen can also, of course, be attained is through excretion by animals. So animal excreta also contain some nitrogen, and that nitrogen could be converted by bacteria and fungi to ammonium again, and the cycle continues. And also lightning fixes nitrogen as well. When it hits the ground, processes occur. I'm not actually aware, I don't really know what the processes are when lightning creates nitrogen, but that is another way that nitrogen can be fixed. And that's a huge energy expensive process, of course, that happens because there's a lot of energy involved. And this is how we create nitrogen, a man-made type of nitrogen fertilizer, through a very energy expensive process. But these bacteria do it for us. They've been doing it for millions of years, feeding plants and then feeding us and other animals as a result. So once again, we can just look at this nitrogen cycle here just to go through different variations of how it looks. Atmospheric nitrogen comes down, nitrogen-fixing prokaryotes convert nitrogen, dinitrogen, to ammonia. So that's the gas form, to the ammonia molecule. And that can be converted by nitrifying prokaryotes to nitrites or nitrates. So the ammonia is converted by nitrifying bacteria to nitrates and nitrites, and that can either go directly to a plant or denitrifying prokaryotes convert it back into nitrogen gas that goes back into the atmosphere, and the cycle continues. But once again, we want these molecules to stay and cycle throughout the soil once made. So nitrogen cycle relies on nitrogen gas conversion into usable ammonia nitrates or nitrites, because these are plant available. Ammonification is organic nitrogen in organism excretions and dead organisms in the form of amino acids and DNA, and moving on to becoming inorganic ammonia. So that's what ammonification is conversion of ammonia from organic nitrogen that is present in organisms into ammonia. Nitrification is the oxidation of reduced nitrogen, so the ammonia turning into nitrite or nitrate. Denitrification is the reduction of nitrate and nitrite to gas, which is nitrous oxide. That's one form of gas that can be formed, and it's actually it's a global warming gas. And the other form is just dinitrogen. It goes back to where it originally came from. So hopefully that gives you an explanation of the nitrogen cycle. Now, there are different biological ways that nitrogen can be fixed, but both of those are by nitrogen-fixing bacteria. And it happens both in agricultural systems and in natural systems. And it happens in agriculture in crops and in pastures and fodder as well. And that different bacteria might vary across this, but the main idea behind here is that they can be either plant associated, where the plant produces symbiotic nodules for the bacteria to live in, versus free living bacteria that don't require a nodule, just require a little bit reduced oxygen conditions for their enzyme to function without being present in the nodules, but being free living outside of the plant host. Different groups of plants have different groups of organisms doing this, and the same happens in natural systems. So basically, all ecosystems contain plants that have nodule symbiotic bacteria within their nodules, so living inside of the plant, versus free living nitrogen fixers. And this is once again just going sort of deeper into this process where we can see the nodules being formed, nitrogen coming from the air, and nitrogen compounds being fixed by the bacteria within the nodule. And this is the bacteria within the nodule zoomed in on. And then the nitrogen compounds go from that nodule to the plant, and in exchange, photosynthetic products, sugars, are fed to the nodule bacteria. So basically the same cycle, but just involving the plant now on a more deeper level. And so 98 species in 13 genera are diazotrophs. So that means that they feed on dinitrogen. They fix that nitrogen. So 1/3 of global nitrogen demand, which is 70 million metric tons per year, are produced by nitrogen fixing bacteria. And that equates to up to 584 kilograms of nitrogen per hectare per year. So that's a huge addition of nitrogen being given to our crops through these bacterial processes. And this is specifically related to Fabaceae, the family Fabaceae, which are legumes like beans, peanuts, peas, and they contain the most studied symbiotic bacterium called Rhizobium. So we can actually inoculate our plants with this bacterium, and they will form these nodules if conditions are right, and will kickstart the process of nitrogen fixation. And we could call these root rhizobiome because it's the microbial community within the root system. So the rhizobium is part of that community. So dinitrogen goes to ammonia, as we already know, and that becomes amino acids, which then form proteins within the plant and within our bodies as well as we consume amino acids. Nucleotides, so as I said earlier, RNA, DNA, ATP, but also vitamins, flavones, and hormones. And this process actually is required to contain sugar to feed the microbes, to feed the rhizobium, and this is in the form of malate that the plant provides it. And also the oxygen concentration has to be a little bit reduced, hence the nodule. And for the nitrogenase enzyme to do this conversion of dinitrogen into ammonia, and iron-containing protein leg hemoglobin, similar to that of our blood, actually reduces that oxygen within the nodule. It basically chelates it. It holds on to that oxygen and removes it from the environment so the nitrogenase enzymes can do the fixing of the nitrogen for the plant. This is another version of showing you exactly how this happens. We've got nitrogen gas, nitrogenase enzyme goes into ammonia and forms amino acids. And this is happening within the rhizobia bacteria in legumes. So this is in the cell of the root within the nodule. And these amino acids are then used to create nucleotides, vitamins, flavones, hormones, and that goes into the legume plant, which we then consume, of course. And then carbon dioxide is released, of course, as well, because everything's living, and that carbon dioxide is fixed into organic acids by photosynthesis, and those organic acids or sugars that are produced by the plant are given to the nitrogen fixing bacteria. And nodulation is quite an interesting process, and you can see how lovely and pink they are with the leg hemoglobin molecule. That's why they're lovely and pink, and we know that they're active when these nodules are like this beautiful color. But exuded flavonoids, polyphenolic compounds, are implicated in this process. So the plant provides these flavonoids to attract rhizobacteria, and this allows for the bacteria to actually express nod genes, and that becomes converted into a nod factors. And these nod factors allow root hair curling around the bacteria to start occurring. So they basically modulate the plant behavior and allow the bacteria to start having a place to live around, first of all, the root, but then they move into the root. So an infection thread starts forming, and this is to allow the rhizobium to enter epidermal cells, and cell division starts occurring. Once cell division occurs, nodules are formed, and the root hair encapsulates the bacteria, forming microcolonies. So it literally just winds around like a snail, and the bacteria get stuck inside, and they enter the nodule via the infection thread into the cortex. So an infection thread is formed as well within this process, and what this means is that the bacteria can actually enter into the root cortex. It allows, they transfer around the plant cells. The bacteria then become surrounded by the symbiosome membrane. So basically they get protected in this membrane, this lovely membrane that forms around the bacteroids. The bacteroids are bacteria after they have their cell walls stripped off. They basically change in shape and change in function the moment they become symbiotic. So these bacteroids then fix the nitrogen. This process is actually affected by heat, acidic soils, drought, and nitrate application. So it's a very sensitive process and a very beautiful symbiotic process that plants and the rhizobia bacteria go through to fix nitrogen. There are also free-living nitrogen fixers, so known rhizobial nodule bacteria. So basically, there's no nodules being formed, and these fix nitrogen and also solubilize phosphorus as well and increase plant growth. Mostly there are commensals, so that means that food is supplied to them by internal or external host environment, and they do not form close host association like the nodule bacteria do. The rhizosphere soil provides 36 kilograms of nitrogen per hectare per year due to these free-living nitrogen-fixing bacteria. And they need oxidized organic molecules from decomposition. So they rely on other organisms, other bacteria, to oxidize organic matter in order to obtain enough energy for nitrogen fixation. They also have nitrogenase enzyme, which is also sensitive to oxygen, and these organisms are called microaerophiles, so they only want reduced oxygen conditions. And these bacteria actually rely on having aggregates to be able to have this reduced oxygen condition. So they rely on places around the root where oxygen conditions are reduced, and that usually happens in microaggregates. And lack of organic carbon limits these free-living nitrogen fixers. In experiments, wheat stubble and reduced tillage has been shown to produce 20 kilograms of nitrogen per hectare per year, and that's up to 50% of what's actually required by plants or to produce a good crop. So you can see that these organisms really do deliver nitrogen to the plants and really help us in producing crops. These are the different bacteria that actually have this capacity to fix nitrogen, and they're the free-living nitrogen fixers, just to show you how many there are that have been studied so far. Now, there's also nitrogen fixation in tuberculate mycorrhizae. So I'll just give you one example to explain what this means. Majority of plants have some mycorrhizal fungi on them, and that's because they require nutrients that the plant can't access through the root system itself. And when nutrient is limited, for example, like in this case, where lodgepole pine is growing on an escarpment, is growing in rock, the only way to receive nutrients is through having mycorrhizal or symbiotic fungi on their roots in order to access phosphorus, nitrogen, and other minerals. And so in this case, Suillus tomentosus is one of the fungi that's growing on the roots of this lodgepole pine, and they basically gain these nutrients for the plant. However, this blue staining slippery jack and lodgepole pine, which is Pinus contorta, have been shown to produce tubercules with nitrogen fixing bacteria. So this is very interesting, and you can see this actually here in the diagram. Sort of a kind of a tubercle is formed, and the nitrogen fixing bacteria are inside, and this is what it looks like, a cross section of it. And you can actually see them, they almost look like nodules as well, but we call them tubercules. So this is another interesting multi-level way that plants partner up with organisms, in this case fungi and bacteria, to gain even more nitrogen in nitrogen poor soils. So the tubercle is a densely packed cluster of root tips enveloped by thick hyphal sheath. So the tubercle is basically produced through the interaction of the bacteria and the fungal hyphae, the fungal cells. It is the fungus that produces this sheath to envelope the bacteria into a tubercle and help them to produce nitrogen. And these are just the different groups of bacteria that fix nitrogen within this symbiosis. Arbuscular mycorrhizal fungi and rhizobium or bradyrhizobium inoculation. So if you put arbuscular mycorrhizal fungi, which is a different type to this sort of a system, and nitrogen fixing bacteria, when they're inoculated together, there's even a more of a nodulation process that goes on, bigger nodules, better nodules, greater nodule biomass, greater plant growth, nitrogen fixation, versus if the bacteria are just alone. So just to show you the complexity, that it's not just the plant and the bacteria, but actually fungi play a role in this nitrogen fixation process as well, or can do so. So it's a very, very interesting way of looking at the plant as a multi-organism system that functions in symbiosis with various organisms. So we've just covered the nitrogen cycle, and we're going to move on to the phosphorus, sulfur, and potassium cycle now. And these occur through solubilizing bacteria. So obviously phosphate, sulfur, and potassium are not soluble, and this requires solubilizing bacteria to make them into a plant available form. So we'll talk about phosphate solubilizing bacteria, which are also called phosphate solubilizing microbes, sulfur solubilizing bacteria, and potassium solubilizing bacteria. All right, so let's look at these individually. So phosphate solubilizing bacteria or microbes. As you can see from the table there, there's quite a few of them that have been so far identified. And even though phosphate is an abundant organic and inorganic mineral, it is actually rapidly fixed and has a very low availability. It is a non-renewable, scarce resource, and we probably have up to 100 years left if we keep mining it. And 80% of total phosphorus in most soils is in this organic form, which is actually insoluble and unavailable. So phosphate solubilizing microbes convert it into orthophosphate, which makes it available to the plant. And there's a few bacilli and pseudomonas and rhizobium even that can do this. So it's a required element for nodule initiation and development and functioning. So it actually plays a role in nodule formation and nitrogen fixation. And if the phosphorus is made available by the solubilizing bacteria or microbes, its application can be reduced by 50%. So we don't have to keep putting phosphate into our agro ecosystems. The thing is also that, as I have stated right at the beginning of the course, is it's actually not very limiting nutrient. It's just that it's unavailable because the microbes are not there to actually dislodge it from the mineral or organic component from the soil and provide it to the plants. There are several phosphate solubilizing microbes. The reason why we say microbes is because it's not just bacteria that can do this, also fungi can solubilize phosphate. But these hydrolyze organic phosphate through extracellular enzymes such as phosphatases and phytases, and they transfer phosphate between the inorganic and organic pools. So they can either add this phosphate to be mobilized or release it where it becomes available to the plant. So phosphate uptake by avascular mycorrhizal fungi is also possible, and avascular mycorrhizal fungi actually play a huge role in agricultural and other systems where they make phosphate available to plants. So let's look at sulfur now. Sulfur is another limiting nutrient, and I should have said that limiting nutrients are those that are not available for plant growth but are absolutely essential, but not easily available to the plant. Our bodies are similar to that of plants, that there are minerals that we can't manufacture by ourselves in our own bodies, so therefore we're required to eat plant material to actually supplement these nutrients that are taken from outside of the body and then converted into available forms in our body by microbes as well, majority of the times, or enzymes. So it's the same that happens in plants. So sulfur is also an essential limiting macroelement, and it is actually being reduced in its atmospheric concentrations. Its levels are dropping, and it is rapidly removed by crops. So it's essentially being mined as crops are being removed. We're removing it through cropping. So it is an integral part of proteinaceous amino acids such as cysteine and methionine, and non-protein amino acids such as lanthionine and ethionine. So these are all just chemical terms that we're throwing around here. Tripeptide glutathione also requires sulfur as well, and this is a very important molecule in detoxification in organisms. Vitamins are also needing to have sulfur in them as well. So thiamine and biotin, for example, are two vitamins that require sulfur. Also phytokeratins, chlorophyll. These are all things that you can look up for yourself if you don't understand what all these mean. They're all just parts, basically, our enzymes and molecules within our bodies and plant bodies that need to be produced in order for them to function. Sulfur also plays a structural role in cells. Disulfide bonds in proteins are formed in order for amino acids to become proteins. Enzymes are being regulated where sulfur is involved, which is a redox control. So this is also, once again, redox reactions function in all kinds of essential biochemical components of plants and animals. Oxidative stress protection is given through the glutathione and derivatives of that as well. For example, in heavy metal detoxification out of a plant or out of human body even as well. Disease protection and defense. Glucosinolates are formed, and allen, and these are compounds that obviously protect versus disease. Sulfur also prevents fungal infections, and some plants actually accumulate or store sulfur in the xylem tissue where the water is transported within the plant. They actually accumulate that and protect the plant from fungal infections. Sulfur deficiency means that there is a reduction and alteration of root exudates, and this impacts bacterial communities. So bacteria around the root zone are there to actually provide that sulfur, but if there's a deficiency in these microbes, this actually prevents other microbial processes as the communities of microbes or the biodiversity is diminished. Sulfur solubilizing bacteria are very important, therefore, in soil. And there's a cycle here that you can look at on the left here in the diagram in more detail to understand how it all works, but it's quite complex. And so I'm just going to explain the basics of what happens between the bacteria and the plant in how sulfur solubilizing bacteria promote plant growth. Over 95% of soil sulfur is in an organic form, and sulfate esters and sulfonates. Sulfonates are the dominant form of sulfur, and these are biological deposition and humidification products, so they're not available to plants. And fungi and bacteria are able to get sulfur from sulfate esters using sulfatase enzymes, sulfatases, and bacterial multi-component monooxygenase system is something that allows sulfur to be gained from sulfonates in bacteria. And these are just a few names of bacteria like Rhodococcus, for example, taxa that are able to actually do this solubilizing of sulfur. Some of these bacteria have genes that are specific to sulfur solubilizing capabilities and enzyme production that allows them to remove sulfur from complex organocompounds. Sulfur cycling is a complex interaction of several free-living and symbiotic root-associated microbes. So that's why it's a very complex process. One organism will modulate the sulfur compound in a particular way, and then that has to be further modulated by other microbes. So rhizosphere and mycorrhizae bacterial attachment to hyphae allow organosulfur mobilization. So fungi on the roots, symbiotic fungi within the rhizosphere of plants, have specific places where bacteria can attach to the hyphae and then allow mobilization of the organic sulfur. So they work together as a symbiotic relationship to support plant growth. A vascular mycorrhizal fungi, their hyphosphere increases bacterial abundance and activity and leads to aromatic sulfonate desulfurizing bacteria. So it promotes the activity of these bacteria. So the hyphosphere, where the hyphae is the fungal cells on the roots, they allow this abundance and activity of these particular bacteria that can desulfurize organic material and from aromatic compounds access the sulfur for the plant. So it's a bit complex, but if you want to look at this in more detail, there's several papers that I've listed in the references and you can have a look at this. But the bottom line is that without bacteria and fungi, and especially in this case we're talking about solubilizing bacteria, sulfur, potassium and phosphate are not available to plants. So this is the key thing we're trying to get across, that these organisms are so totally essential in the functioning of a healthy plant ecosystem, and the soil is rich in all of these minerals. However, they are not available to plants unless the bacteria are present. So the last cycle we want to look at, or the last mineral that we want to look at in terms of being also limiting and essential constituent in all living cells, is potassium. So potassium solubilizing bacteria promote plant growth, metabolism and development. And this particular element is also essential in immunity and abiotic stress reduction in plants. So it activates over 80 enzymes and provides energy metabolism, starch synthesis, nitrate reduction, photosynthesis, sugar degradation. So very, very important in how the plant functions. Potassium is absolutely necessary. This is the concept of adding the NPK to the soil. And of course, none of this needs to happen if we have the potassium solubilizing bacteria. It is the most abundant soil nutrient and in fact the 7th most abundant element on Earth. So once again, we don't need to add a synthetic variant of this to soil. 90 to 98% of the mineral is actually unavailable to plants and causes deficiency. And fertilizer is actually quite expensive and polluting, but potassium solubilizing bacteria make the potassium available to the plant. They solubilize that mineral from the soil. Potassium solubilizing bacteria are actually present in all soils, and their numbers, diversity, and ability to solubilize potassium vary with soil and climatic conditions, but I would say and add that it's mostly to do with the soil ecosystem and the availability of these bacteria, as well as the biodiversity of these bacteria. So once again, we just need to provide the conditions within a soil ecosystem to support the proliferation of these very essential organisms. Potassium solubilizing bacteria have several mechanisms by which they are able to dislodge potassium from organic molecules and from the mineral component of the soil. These are just the different species of bacteria that are able to gain potassium from biotite, feldspar, elite, muscovite, orthoclase, and mica, and these are all just clays. So obviously these types of clays contain potassium, and these bacterial species can remove that potassium from these clay mineral complexes. They use organic and inorganic acids and the process of acidolysis, which is explained here, and complexolysis, which is explained here. And also they use polysaccharides, so these are some things we'll talk about in the next section, and chelation, where they can just create molecules that absorb the potassium to itself and then remove it from a bigger complex. And of course, they produce all kinds of exchange reactions. So for example, hydrogen ion, when it's removed, when it becomes free, when it's dislodged with the addition of water and it forms acid, that hydrogen ion can actually displace potassium, magnesium, calcium, and manganese and make that available to the plant. And and this also allows for different metals to chelate that hydrogen that went into solution, and this is complexolysis. These are two things you can just look up if you're more interested in the chemistry. It's quite complex, but it's quite fascinating to understand how these bacteria actually do these jobs. So seed and plant inoculation experiments show that when there's increased potassium uptake via these bacteria, germination is increased, plant vigor, plant growth, and yield. When tomato plants are grown with silicate-dissolving Bacillus mucilaginosus, the plants produce 125% greater biomass, contain 150 times more potassium, and also phosphate uptake is increased in these plants as well. But despite this research, the uptake of this science is quite slow, and I would suggest that even though that these are isolates of bacteria that have been studied and are showing promising results, I am not proposing that we should be in isolation adding these to our soils in order to increase potassium availability. What we need to do is create an ecosystem for these organisms to actually proliferate in our soils. So really what we need to be doing is just increasing soil biodiversity and stopping the various methods by which we reduce that biodiversity through applying fertilizers and tillage and doing all these destructive things like applying chemicals to kill weeds and all kinds of things that actually destroy these organisms because they naturally should be present. And in forest ecosystems, obviously this functions really well and there are no potassium or any other mineral deficiency. So this is the end of lesson 3.1 and we're going to move on to lesson 3.2. In lesson 3.2, we'll talk about plant growth promoting rhizobacteria, extracellular polymeric substances that bacteria produce, and the process of rhizophagy. So I'll see you in the next section of lesson 3.